All right.
Pay attention to what's coming up right now.
So, nucleus. So we're talking about all of our cell organelles. Nucleus, one of the most obvious organelles as you're looking at a cell. It's one of the largest organelles. It generally stains using iodine or methylene blue, stains darker than the surrounding cellular material. And the nucleus contains some important things. So it contains the genetic information in the form of chromatin. So there's some terms that are similar to each other. And we'll get more to this when we talk about cellular reproduction and the details of mitosis and meiosis, and when we talk about genetics. But the DNA, DNA is long molecule, deoxyribonucleic acid, and in most living things, it is sort of wrapped around a structure of protein. And we call that material chromatin. And when a cell is just sort of under its normal cell cycle and just doing the normal functions it needs to complete, that chromatin is sort of all unwound and spread throughout the nucleus, sort of thread-like, all spread throughout the nucleus. When it's time for the cell to actually reproduce and divide, that chromatin kind of winds up into what we call chromosomes, which you may remember those X-shaped things. Those are little bundles of DNA. So the chromatin winds into chromosomes when a cell is going to divide. But that's the genetic material. Here you see it all unwound inside of the nucleus. The DNA basically directs all of the cellular activity of the cell, contains all the genetic information. It codes for all of the proteins and traits that make up that cell. And every cell basically contains the same DNA. Each of your body cells with a nucleus has all the instructions on how to build you. Every single cell. All cells contain the same DNA. The outer layer of the nucleus is called the nuclear membrane, similar in some ways to the cell membrane. It's a double layer. Okay. It's semi-permeable. And it has pores in it, these little openings, nuclear pores, because certain things can move in and out of the nucleus, other things cannot. For example, the chromosomes, the DNA is stuck inside of the nucleus and cannot exit the nucleus. But smaller molecules, things like messenger RNA, they can get out of the nucleus through those nuclear pores. And they actually, their job, and we'll talk more in detail about this, is to sort of shuttle the information from the DNA where it's stored inside the nucleus out into the cytoplasm to the ribosomes. They bring that information back and forth, these molecules. Also inside of the nucleus is the nucleolus. Okay? The nucleolus is made of a combination of DNA and RNA and proteins, and its job is to synthesize the ribosomes. Okay? And ribosomes are important in the making of protein, synthesizing protein. Some cells have multiple nucleoluses, and generally when the cell is about to divide, the nucleolus disappears and then reappears after cell division is over. So here you see an electron micrograph showing this is the nucleus internally. You can see the nuclear membrane, and you can actually even see the pore, this little opening in that nuclear membrane. The ER, Endoplasmic Reticulum. The endoplasmic reticulum is basically a series of membranes, okay, and that do several things. They help to transport things within the cell. They also help synthesize or make different materials, sometimes proteins, sometimes other substances. And what you see when you look at the ER, they generally are close to the nucleus. And there's a series of windy sort of passageways called the cisternae. And this windy area provides for a large amount of surface area for various chemical reactions to take place. Okay. Also, there's two types of ER, rough ER and smooth ER. The rough ER, when you look at it in the microscope, it looks like it's dotted with all these little circular organelles. You see in this electron micrograph, here we have the nucleus, and here's the rough ER. You can see all these spots on them. You can see it here. Those little spots are ribosomes, sort of attached to the rough ER. And the rough ER, because it has these ribosomes on it, is responsible for making proteins. So the ribosomes actually synthesize the protein. That happens in conjunction with the rough ER. The smooth ER also can synthesize other non-protein molecules. So that's the smooth ER is out here. It doesn't have those ribosomes attached. So these ribosomes are these small, they're sometimes not called organelles. A lot of times we call organelles things that are surrounded by a membrane. And the ribosome is not surrounded by a membrane. It's actually a little chunk of RNA. And the ribosome's purpose is to synthesize proteins. They take instructions from the DNA and they assemble proteins. Some of these ribosomes are attached to the ER. Okay, you can see these attached ribosomes. Others are free and just sort of floating in the cytoplasm. Usually the ones that are attached are used to synthesize proteins that are going to be excreted from the cell, that are going to leave the cell, whereas free ribosomes are used to synthesize proteins that are going to be used within the cell. And this is an RNA structure. Has different subunits to it. It works with other types of RNA, transfer RNA and messenger RNA, to assemble proteins. So what you see here, this whole thing is a ribosome. Okay? This messenger RNA contains the instructions about how to build a protein. It came from the nucleus. This transfer RNA sort of reads this messenger RNA and brings in amino acids to create what's going to be a protein eventually. So these types of RNA work together to synthesize these proteins according to instructions from the cell. Mitochondria. If you remember from 7th grade, what do mitochondria do? Cellular respiration. They release energy. What is the molecule that serves as sort of the energy currency in a cell, Sophia? Almost. ATP, adenosine triphosphate. That's what our cells use as energy. And so ATP is released by the mitochondria. They're usually shaped like a sphere or a rod. Okay? They also have a double membrane. And within that membrane, they have all these folds. Okay? They're called cristae. And they serve to, again, increase surface area. Whenever you have like folded surfaces, you're increasing surface area. Just like in our intestines or our lungs, you have folds and complex shapes because they increase surface area for chemical reactions to take place. Mitochondria actually contains its own DNA because where do scientists believe mitochondria evolved from?
Heterotrophic cell.
Yeah, what kind? Aerobic bacteria which use oxygen. Okay, and that's where they feel that those mitochondria evolved from. So they have their own DNA; they can replicate themselves. Usually, cells contain varying numbers of mitochondria. Depends on the type of cell. Typically 300 to 800, but in cells like muscle cells, which require a lot of energy for the constant contractions, they would have more mitochondria. The number of mitochondria in cells can change over time. That as it's one of the adaptations as you exercise. Okay, is that your cells adapt to that exercise, respond to it. by producing more mitochondria, allowing your muscle cells to work more efficiently. As cells age, the number of mitochondria decreases. And here again is an electron micrograph of a single mitochondria, where you have sort of the inner space is called the matrix, and the folds are called the cristae, and they're all contained within this double membrane. Golgi bodies.
Golgi Bodies.
Yep, Golgi bodies. Sometimes they're called Golgi apparatus. Sometimes Golgi complex. They're all the same thing, but the Golgi bodies are a part of the cell. They look sometimes similar to the ER, in that they're a series of membranes, kind of a flattened series of membranes. And their job is to prepare a material that's going to be secreted from the cell. What does it mean when I use that term, secreted?
Like when it leaves the cell?
Yeah, released from the cell. And so these series of membranes, they take materials that were produced in the ER. And so if you look at this, there's two faces. There's called the cis and the trans face. The cis face sort of faces the ER. And so materials from the ER come into the Golgi bodies. That's a vesicle, a little sphere of material from the ER. They merge with the Golgi bodies, where these materials can be packaged up and then sent into new vesicles, new little bubbles that get sent and eventually are going to leave the cell. So these products are on their way to leave the cell. They're created in the rough ER, sent to the Golgi bodies to be packaged up, and then sent out to eventually leave the cell. And again, you see the flattened series of discs. Anyone ever listen to the band Fish?
What?
Fish, the band Fish. Yeah, they have songs references several cell organelles, including Golgi apparatus and lysosomes and others. So it's a song to listen to.
Band.
Maybe I'll play it for you at the Sunday class here in the jam band. So cell parts. Lysosome, another organelle. Lysosomes are generally small organelles, and their job is to help with the breakdown of materials. They contain enzymes in them, hydrolytic digestive enzymes, enzymes that are involved in the breakdown of material. And so they are used for several functions. When cells have to destroy a pathogen, like a bacteria, okay, they engulf that bacteria, and then lysosomes release those digestive enzymes to break down that pathogen. In unicellular organisms like paramecium, after they ingest food and it gets stored in a little food vacuole, the lysosome merges with the food vacuole, releases its enzymes to break down that food. When cell organelles are no longer functioning, cell organelles sort of age and need to be broken down eventually. The lysosome can do that. Okay. In the tadpole, for example, as well, when the tadpole is reabsorbing its tail as it metamorphs into a frog, lysosomes are involved in that process of breaking down that tissue that made the tail. They look just like a small little sort of sac that has enzymes in it. I mean, you wouldn't be able to actually see the enzymes. This shows them as little dots. They're also rarely found in plant cells, okay? So they're mostly just found in animal cells. And you see here the ingestion of food into a food vacuole, the merging of the food vacuole with the lysosome to digest that material, and then any materials can be eliminated from the cell. Vacuoles. Vacuoles are storage areas. They store various cell products, water, food, the surrounded by a membrane. They're specialized sometimes in unicellular animals, things like an amoeba or paramecium. There's a food vacuole. So as the paramecium ingests food into the oral groove, it goes down the gullet and ends up in a little bubble that we call a food vacuole. Stores the food. Also, no, it's not a video. The single-celled organisms have contractile vacuoles. And we saw these in the protist lab where they bring in water because of osmosis. These organisms are constantly taking in water. And so to eliminate that excess water, these contractile vacuoles basically pump it out, squeeze it out. But it's another type of vacuole. Plant cells have large central vacuoles. Okay, you can see one here. You can see an actual electron micrograph of one here. And basically that fills up with water and maintains the rigidity of that cell. Because as that central vacuole fills up, it puts pressure on the cell wall and keeps it rigid. Gives it its shape. It's why plants wilt if you don't water them, because those central vacuoles, the plant doesn't have enough water, the central vacuoles lose water, and they're not exerting that pressure, keeping the plant upright, and it starts to sort of droop over. And then our last organelles are involved in what we call the cytoskeleton. Centriole is an organelle or a cell structure that is involved in the division of the cell. It also is important in forming cilia or flagella in cells that have those parts. Centriole is involved in pulling apart the chromosomes during mitosis and meiosis. And it's generally found in an area near the nucleus. The area itself is called the centrosome. There's usually two of them, and they're paired, and they're at right angles to each other. And they're made of these small, we're going to learn about in a minute, microtubules, these little molecules that form these tubes. The cell wall gives structure to a cell. It's a non-living layer. It's outside of the cell membrane. And it gives cells a rigid shape, a defined shape. It has small openings in it to allow material to move through it. It's non-living. It doesn't participate in any metabolic processes or any sort of transport. It's basically just a non-living outer shell. And it can be made of different types of materials depending on the type of organism. Plants and algae have cellulose making up most of their cell wall. Fungi have chitin. Bacteria have peptidoglycan. So there can be various materials that make up this cell wall. Not found in animal cells. Chloroplasts. Chloroplasts, here you see an actual electron microscope image of the chloroplast, these small little green circles within a cell. What happens there in the chloroplast? Photosynthesis. This is where photosynthesis happens in plants and protists. They contain chlorophyll. That's the green pigment that makes chloroplast green, that makes leaves green. They're a type of what we call a plastid, which is a membrane-bound organelle that has various pigments. And the chloroplast looks like sort of a disc shape, if you look closely, and it has these stacks of sort of discs that contain chlorophyll, thylakoids called, and these stacks are called the grana. And then the space around them is called the stroma. It's a double layer, an inner and outer membrane. Here's an actual electron micrograph image of what a chloroplast looks like. Again, chloroplasts, like mitochondria, have their own DNA. And again, why is that? What's the significance of chloroplasts having their own DNA? Cormac?
Because they're supposed to be like digested anaerobic bacteria.
Not anaerobic bacteria.
Anaerobic?
No. Think about what the chloroplast does. Yes? They're autotrophic bacteria. Well, autotrophic bacteria, I mean, saying anaerobic or aerobic is referring to respiration. But these are referring to their photosynthesis. So they came from photosynthetic bacteria, okay, which are also can aerobically go through respiration to release the energy from that glucose. All right, almost there. Microtubules, microfilaments, these are parts of the cytoskeleton, sort of the internal structure of a cell that allows for the structure for things to move. Microtubules are, just as the name would suggest, hollow little cylinders, a tube that gives the cell shape. They're found in the cilia and the flagellum and centrioles. They're made of special types of proteins called tubulin that arrange themselves in a helix, which forms a little hollow tube. And there's also another type of material called microfilaments, which are thinner, thread-like structures. These structures can contract, and they're very important in the movement of muscle tissue. Okay, they're made of material called actin, which can contract and relax. And so they have there are many microfilaments in skeletal muscle. And finally, related to these microtubules and microfilaments are some cell organelles that are specialized and found in specific types of cells. Flagellum, is sort of a tail, a whip-like tail. It contains many microtubules. It's used for locomotion in some protists like Euglena. Also, sperm cells of a flagellum that they use for movement. You can see there's this little tiny cellular motor that makes the flagellum move. Okay? And then there's cilia, which basically are identical to a flagellum. They're just different in their length. In cilia, generally there's more of them. Okay? And they can be used for movement as well, like in a paramecium is covered in cilia that sort of paddle it around. But they're also used in certain types of cells. In our trachea, the cells of our trachea, we have cilia, which helps filter out environmental contaminants like dust and things like that. Also, fallopian tubes and the reproductive system in women have cilia that move the egg through the fallopian tube to the uterus after ovulation. And so there are cilia in various types of cells. This is a scanning electron microscope image. You can see these tiny little hairs. This is a transmission electron microscope image where you can sort of see a cross section of those in the flagellum. All right, and that is the cell organelles.
That was a lot to learn.
There was a lot to learn.
Probably not